guys, welcome to your Friday Live lesson. This is Mrs. Kohler. Last week we were talking about mitosis and I gave you a demonstration using the beads and the paper plates showing you how mitosis worked. This week, since we talked about meiosis yesterday, I'm going to give you a demonstration about meiosis, once again using the plates and the pop beads. So we're going to move on to the document camp so that you can see visually how meiosis works. Okay, so just like last week, we have a plate that is signifying our cell. This is our parent cell, but remember, this is meiosis, not mitosis. So we're gonna work with a little bit more DNA this time than we did last time so that I can show you the homologous chromosomes and that I can show you crossing over. So this, once again, is your nuclear membrane. And next to your nuclear membrane, you have a set of centrioles. And inside this nuclear membrane, last week we used 3 strands of DNA. This week we're going to use 6 strands of DNA. So our nucleus is going to look a little bit more crowded, but that's going to clear out pretty soon so that we can organize and see what's going on. So right now we are in interphase of meiosis. We are in G1 of interphase of meiosis. So you have your parent cell, you have your nuclear membrane, you have your DNA inside in the form of chromatin, and you have your centrioles. Next thing that's going to happen is S phase of interphase. So you're going to get another copy of each of these DNA strands. So this is making a pretty crowded nucleus. This nucleus is pretty crowded, and it's still chromatin, and it's still just unwound and in here messily. During G2 of interphase, you are going to have a second set of centrioles be produced, just as what happened in G2 of mitosis. So far we're pretty similar. We're just using a little bit more DNA. Now we are entering prophase 1 of meiosis because remember meiosis happens in 2 phases and it's going to be producing 4 daughter cells instead of 2. So now we are in prophase 1. So a lot of the same things are going to happen. Your centrioles are going to migrate to the poles just like they did before. Your nuclear membrane is going to disappear just like before, and your chromosomes are going to form. So as I make up these chromosomes, one thing you'll notice is my red chromosome and my purple chromosome are the same length because these are homologous chromosomes. So same thing with my green and my orange, they're homologous chromosomes, and my 2 different colors of blue are also homologous chromosomes. So they're pairing up. Now, this is where you have your first major difference between mitosis and meiosis. These are going to line up, these homologous chromosomes are going to pair up and form tetrads. Remember, tetra is 4, so now you have 4 sister chromatids. So these tetrads are pairing up together, and you're going to have crossing over occur. So crossing over is when the arms of the chromosomes that touch each other in a tetrad trade some DNA. So the purple, because the purple and the red are homologous chromosomes, they are going to perform crossing over. So some of the DNA is going to swap within this tetrad. So now you have 4 genetically different sister chromatids that are going to be going through meiosis. So we're going to cross over here with the green and the orange as well. So you can see that the green and the orange traded. And now the two colors of blue are going to trade with each other. All right, so now that crossing over has occurred, you have 3 tetrads. You have this tetrad here, where the inner 2 arms of the chromatids traded and did crossing over. You have your blue tetrad here, where the inner 2 arms did crossing over, and you have your other tetrad here, where the inner 2 arms did crossing over. Notice that the outer arms don't cross over. They're only crossing over where they touch. Crossing over only occurs on one of the sets of sister chromatids. So in this stage, the nuclear membrane disappeared, the centrioles migrated, and your tetrads formed and crossing over occurred. Okay, now you're going to move on to metaphase 1, where these tetrads are going to line up along the middle, because remember, M for middle. So you're going to get this tetrad here, this tetrad here, and this tetrad here. So you have the 3 tetrads lining up along the middle. Metaphase 1. Anaphase 1, the tetrads are going to pull apart, and an entire chromosome is going to go this way, an entire chromosome is going to go this way, an entire chromosome is going to go this way. So where in mitosis the sister chromatids were separating, here the homologous chromosomes are separating and the tetrads are splitting up. Anaphase 1. And then telophase 1, these are going to group back up, and the DNA is going to relax a little bit. It's not going to completely relax back into chromatin, but it is going to relax a little bit. And you're going to get a nuclear membrane forming around each end with centrioles hanging out at the side here. That is telophase 1. So 2 nuclei, 1 cell. And then your cytokinesis is going to occur. So this nucleus is going to move over here. Now, meiosis 1 is complete, but you still need meiosis 2 to happen because your cell hasn't gone yet to haploid. You still have diploid cells. So the cells at this point, these are 2 daughter cells. At this point, they're going to enter a period called interkinesis. During interkinesis, the DNA is relaxed a little bit. But it's sort of just a brief resting period between the 2. But one thing that is going to happen is you're going to get another set of centrioles in each cell. So that was interkinesis. Now, for the next few stages, there's going to be the same thing happening in 2 cells. So this is meiosis 2, because it's the second stage, but also because there's 2 of each cell. So now we're going to do prophase 2. So same thing that happens in prophase, the centrioles are going to migrate to either end, but because it's prophase 2, it's happening in 2 cells. Nuclear membrane's going to disappear in both of them. And your chromosomes are going to form up. Now here is one way that you can tell a difference between this happening between mitosis and meiosis. There's actually two different ways to tell. Number one is that it's happening in two cells at the same time. And the second way is that you can see that these sister chromatids are no longer identical. They are genetically unique. So you can tell this is during meiosis because the chromatids are a little bit different from each other because of that crossing over that occurred. So that's prophase 2. Metaphase 2, we're going to line up along the center, but it's happening in 2 cells at the same time. Move this so you can see the centrioles. So these line up along the middle. Metaphase 2, and it's happening with 2 cells. Anaphase 2, your sister chromatids are going to separate. And this is going to happen on both sides. So because it's happening in 2 cells, you know it's anaphase 2. And because they're pulling apart, A for apart, A for ana, anaphase 2. Telophase 2. This is going to, you're going to have the nuclear membrane reform around each one. So you're going to have 2 nuclei inside each of the daughter cells. So because this is happening in 2 cells, you know that it's part of meiosis 2. So, and you have 2 nuclei in each one, so telophase 2. Can't see the centrioles, but they're here. They keep slipping underneath. All right, so this is telophase 2. And then cytokinesis is going to occur in each of these. So the cells cleave apart. You have your cleavage furrow. And 1 nucleus goes into each new cell. I'm just going to put this nucleus where you can see it. And over here, 1 nucleus goes into each new cell. And also, if you look at the chromosomes in each of these, they are slightly different. So I'm going to not make them so chromatin-y. I'm going to make them so that you can see them clearly. So you can see that each one of these has genetically unique composition. When you look at the colors are not the same in any of the 2 daughter cells that are produced. That is because meiosis produces genetically unique daughter cells. They are different from each other. So when this occurs, that means every sperm and every egg that is created is different from every other sperm and every other egg, which is how, even though you have brothers and sisters that came from the same parents, you're all very different people because you all got a different assortment of genes. So you now have 4 haploid cells that are each genetically unique. Thanks for joining me for this journey through meiosis. I hope you enjoyed the theme music again, and I hope you have a better idea how it is that meiosis happens after having watched this video. Thanks for joining me, and see you in class. Metaphase 1 is complete, but